Okay, good morning. Today is round 2. For people who are just joining us, I remind you that this is a study that's jointly supported by all 3 academies, science, engineering, medicine, and I also want to reiterate my thanks to the Moore Foundation for having supported the travel grant so that the future of this field gets to experience some of the programs we've generated. Today we're going to meet through noon as an open session, and we'll be very much like yesterday in that we'll be hearing from experts, and there will be time for questions and talk. After lunch, the public part of this meeting will adjourn, and the members of the study committee will meet in closed session to talk about the final report that's going to result from the various inputs we've had. Whenever a group of people who know a lot about their individual fields gets together and tries to create a committee document, it's a struggle and an opportunity. I sometimes like to joke and say that, teamwork means never having to take the blame yourself. And we have been charged with making some recommendations. I suspect a lot of our recommendations are going to be about prioritizing future research because there are some very big knowledge gaps when it comes to connecting the indoor microbiome to human health, and we've been hearing about them. But without further ado, I'd like to start today's first session entitled Beyond Bacteria, Viral and Fungal Ecology in Indoor Environments. And we're going to start with the itty-bitty microbes, the viruses. And so our first speaker is Linsey Marr from Virginia Tech. Viruses in the built environment. Welcome.
Thank you for the invitation to speak here today. I've been following these meetings remotely, so it's nice to be here. I'd like to start out by thanking the people who actually did the work that I'll be talking about today. There's several people in my research group, students and postdocs, and Dr. A.J. Prusson is actually here. He's a research scientist in my group, and my collaborators who are faculty at Virginia Tech and also at University of Pittsburgh. This research has been funded by a combination of NIH, NSF, and the Sloan Foundation, and I mentioned some of our nanotechnology initiatives because our thinking about nanoparticles in the air and in the environment has informed some of the work that I'll be presenting here. My background is actually in air quality engineering, and so we take, so I have this very engineering approach to things where we track the fate and transport of things in the environment, and I've applied that idea to viruses. So with air pollutants, we have a source of emissions, which might be a power plant or factory or motor vehicles. Those emissions then are subject to transport in the atmosphere. This could be indoor atmosphere or outdoor atmosphere. They are also subject to transformations in the atmosphere. These could be particles that coagulate, condensation of gases onto particles, or gas phase chemical reactions, and even heterogeneous reactions. Further transport may occur before we reach some kind of endpoint of interest, whether it's human exposure or deposition of the pollutant on a surface. Now, we can apply this same framework to viruses where we have a source of emissions, which may be someone who's infected and then is coughing or sneezing, and viruses come out in respiratory droplets. And then the types of transformations that can occur to a virus are obviously different from those that happen to chemicals, but there can be biological inactivation due to heat, UV radiation, salt, or changes in pH. And we've identified 8 sources of airborne microbiomes in the built environment. This is not specific to viruses. This is really true for all types of microbes. First, you can have things coming in from outdoor air. We know that humans are a big source of microbes in the built environment, as are pets. Things come in from the heating, ventilation, and air conditioning system. They can be aerosolized from the water into the air from plumbing. We have mold growing on surfaces. We can have plants that give off microbes into the air, and we also have dust resuspension, kind of a secondary source of things that have already settled on the floor. So of the particles that are in the air, there's maybe 1,000 particles per cubic centimeter in the air that we're breathing now. Half of those are probably from resuspended dust. So when people walk or moving, there's resuspending things that have settled onto the floor or other surfaces. Because we're engineers, we always want to quantify things, and so we were interested in understanding, well, how many viruses are actually out there? And so we went and collected samples and there have been some outdoor measurements of estimates of total virus concentrations, but there have not been any estimates indoors. So we went and collected samples in a classroom, a daycare center, a dining facility, a health center, 3 homes, an office, and then outdoors for comparison. And so we actually came up with some numbers, and these are total virus-like particles where we used a fluorescent dye and filtered out the big things and counted the small things. And we estimate that there are several hundred viruses per liter of air in these different settings. And one thing that was surprising was that we expected the concentrations indoors to be much higher than outdoors, because we hear about bacteria and humans as a source, but it turns out that the levels outdoors were about 2 times higher than indoors. And then for comparison, we also looked at bacteria. I know there's kind of this rule of thumb, viruses are maybe 10 times more prevalent than bacteria. Well, our bacteria were present at similar concentrations as were the viruses. Another question that gets at sources is, what is the seasonality of the viruses? And so we went into a daycare center where we grabbed their HVAC filters, their used filters every 2 weeks. We ended up doing 16 srRNA sequencing to look at the bacterial community. We also tried to do viral metagenomics on this, but utterly failed. So we ended up doing PCR for 6 targeted viruses. 3 of them are shown up in that table. We did not detect adenovirus over the whole year, nor rotavirus. We did detect respiratory syncytial virus in the springtime and early summer, and then there at the end of the year in November, December. The seasonality of the disease, it tends to be more active in the kind of late winter and springtime, which correlates with some of our results, but not all of them. One bit of warning is that we analyzed our unexposed filters as a control. This is gets again at the importance of looking at negative controls. We were hoping to look at seasonality of these other 3 viruses. There was that enterovirus 71 outbreak that year. We really wanted influenza A, my personal passion, and rhinovirus, but our blank filters were contaminated or showed the presence of those, so we weren't able to reach any conclusions about those. As was mentioned yesterday, viruses, microbes in the air are subject to transport just like abiotic particles. The physical transport of them is dependent on the size. The size of a naked virus, though, does not tell us how it's going to be transported in the environment, because we rarely have these bare microbes floating around. The viruses are being aerosolized from humans, often from liquid sources, from water, and so they have associated with them water and all the solutes that are carried in the water. Even our ultra pure water that we get in the laboratory has, when we aerosolize it, there's tiny bits and then evaporate off the water. There's tiny bits of salt that are present. So the virus here, influenza, may be 0.1 microns in diameter, but when we're talking about its behavior in the environment, maybe it's associated with an aerosol that is actually 0.5 microns in diameter, and that's how we need to treat its transport. So one of our questions was, well, influenza is thought to be transmitted via the airborne route. Can we actually find it in air in particles that are of a size that would stay suspended in air for long enough? And so we went into, we collected samples in airplanes, a daycare center, and a health center, and we collected them aerosol samples, size resolved. So we separated out using a sampler called a cascade impactor, separated these out in sizes ranging from less than 0.25 microns on up to greater than 2.5 microns. And this shows you the averages in terms of percentage of virions, of virus particles that we found in each size fraction. averaged across our positive samples. And half of our samples were negative, so we didn't find it everywhere. But you can see that 70% of the total virus particles are smaller, associated with aerosols smaller than 2.5 microns, which is kind of EPA's cutoff for what we call fine particles. These and even particles that are a little larger can stay suspended in the air for hours, if not days. So there's certainly the potential for transmission to occur via this route. One of the other kind of transport questions that we've been interested in is resuspension. As I mentioned, maybe half of the particles in the air are indoors are from resuspended dust. And so this came about because I noticed in my family that my husband, who's a foot taller than me, seems to never get sick, whereas the kids and I seem to get sick a lot more often. So I had this hypothesis that he gets to breathe his own clean air that's never contaminated by the rest of us when we're sick. And so you can think about if you're walking on a dusty trail, you're generating all this dust and you see it down near the ground, and some of it works its way upward, but it becomes more dilute and it doesn't get transported as high. So I hypothesized that there would be a vertical concentration gradient in viruses that are resuspended from the floor. So we applied a mass balance model where we took the room, divided it up, and applied a mass balance equation to each vertical slice of the room. In this equation, we have, these are the turbulent transport terms. The second term you're not used to seeing, and that's because, so K is our turbulent diffusivity, and K in this case varies with height. So we actually did some experiments where we had people walking past air velocity meters to see how the turbulent diffusivity varies with height. We found that it's highest actually at the height of your hand, where your hand's swinging. So we needed this parameter in order to put it into our model. And then we have removed, so that would be how the viruses are transported up and down. They're also subject to settling. And then we have the source at the ground. That's due to the person walking and resuspending things from the ground. And here's what we found. So this shows you, as a function of height along the y-axis, just pay attention to the solid line where we have one person walking in our model. The x-axis shows the normalized infectious viruses, normalized to the concentration at 0 meters, or right at the ground, right above the ground surface. So you can see that someone who is maybe 1 meter tall, one of my kids, would be exposed to a concentration that's about 75% of what you would get right at the ground level, but then someone who's 2 meters tall, like my spouse, would be exposed to only maybe 0.67% of that value. So maybe there's a, let's say, 15% difference in terms at those two heights in terms of what the person's being exposed to. So I'm not sure we can really blame his wellness on that entirely, but maybe it factors in. The other thing we've done is to model virus dynamics indoors in considering the room as a well-mixed, what we like to call reactor or well-mixed control volume, where, again, we're applying a mass balance, and the viruses are subject to removal by settling, by ventilation, and by inactivation. So now we're looking at actual infectivity of the viruses. In this equation, C sub D is the concentration of infectious viruses associated with aerosols of a certain diameter d. Now, the settling velocity depends on that diameter d, and the diameter depends on the relative humidity. So when someone coughs, they spit out a certain size distribution of aerosols. Those are then subject to evaporation, and the drier it is, the more they evaporate and the smaller they become. So that can affect the transport. And it turns out that the inactivation rate is also a function of relative humidity. And some of that information is available in the literature, although we're kind of revisiting some of that because there's conflicting results. So now we're looking at the size distribution of these virus-laden aerosols that are from a cough, and we're assuming they're well mixed into the room immediately. We have a room with like a residential room with an air exchange rate of 1 per hour and a relative humidity of 50%. So this shows you that initially, the y-axis shows you the concentration of viruses, infectious viruses. And so when the person initially coughs, we have some information about the size distribution of a cough, and then we assume that the viruses are evenly distributed at a certain concentration per volume. So initially, we have more viruses in these associated with these larger droplets because they have so much more volume. But then things evaporate quickly within under a second. And so a minute later, everything has shifted to the left because those those droplets or aerosols evaporate and shrink, and we also lose the larger ones preferentially by gravitational settling. And as time goes on, we can see that this curve overall tends to shrink down because we're losing things by settling and ventilation, and it's also shifting to the left because the settling preferentially removes the larger particles. As I mentioned, the inactivation rate is a function of relative humidity, and so we applied a certain relationship from the literature to, for our inactivation rate, K. And you can see that, again, this is a size distribution of infectious virus particles, and you can see that it is higher, the red line, at 10% relative humidity than at higher humidities. And that's because, again, the inactivation rate is lower at the concentrations are higher at the lower relative humidities, like the 10–30%, because inactivation is thought to be lower at those humidities. This led us to wondering about, well, why is there this relationship between humidity and viability? Because as I said, it's not bare viruses that are floating around out there. They're actually probably encased in these aerosols, so they're not necessarily even exposed directly to air. Well, it turns out that we can calculate the, so as I mentioned earlier, you cough or sneeze, and these droplets are leaving an environment, your respiratory tract, that is saturated, 100% relative humidity, entering the ambient environment where the humidity here might be 40% or 50% or so, and they're subject to rapid evaporation. So using the Kohler equation, we can predict what is the final diameter of those droplets going to be. So the y-axis here shows you the final over the initial diameter. Notice that the y-axis does not start at 0 nor end at 100%. We're zooming in on a part of this curve. That's where all the action is. So if we start with a droplet that is initially 1 microns in diameter, and let's say that, so you cough or sneeze this out, or it gets aerosolized from water, it then is subject to evaporation, and if the ambient humidity is 10%, we're going to end with a final diameter of 0.4 microns. But even at moderate humidities, say 40 to 60% or 30 to 60% or even 70–80%, which is what we commonly see indoors, we're already shrinking down to less than half the initial diameter. Volume goes with diameter cubed, so that means that we are actually shrinking our volume by a factor of 10 roughly. Any solutes that are in the droplet to begin with are not volatile, salts, proteins, surfactants, and other things, so they're going to be concentrated by a factor of 10. So this led us to a hypothesis, which we're in the midst of trying to dissect further, that relates humidity to as the kind of controlling factor in evaporation, which affects the chemistry of the droplets, which then affects virus viability. And this would apply for bacteria too. So specifically for viruses, though, at low relative humidity, less than, let's say, 50%, complete evaporation occurs. We lose all the water and the solutes crystallize, and they cannot harm the virus. They've crystallized out of solution. And the influenza virus, in this case, can survive under totally dry conditions. At medium relative humidities, we have this, so this is our droplet here in blue, the outline, and then our green and purple dots are the different salts and proteins that may be present. There's our virus. It is subject to evaporation, shrinks by more than half in diameter, and everything you can see down in the lower droplet becomes very concentrated. And so we think those elevated solute concentrations or the change in pH may harm the virus. And we're in the process of, as I mentioned, of exploring this further. So we have generated these droplets, stationary droplets on a hydrophobic slide where we have actually fluorescently labeled the different components of this droplet, and this droplet is simulated respiratory fluid. So it starts out with fairly high concentrations of salt in our protein, which we use as mucin. So we've labeled the mucin with a red fluorescent dye, and then we have a hydrophilic green fluorescent dye that represents the salt. And so you can see that this droplet, which was originally at 100% relative humidity, here we drop the humidity down to 60%, and we actually get this, this aggregation and separation of the mucin, the reddish outline around the outer edge, forming a shell around the kind of the middle part, which is probably still a very salty solution. It can even become supersaturated. And the question is, where is the virus in this droplet, and what is the microenvironment that it's experiencing, which is going to affect its viability or infectivity? And then we think at very high relative humidities, we have minimal evaporation. This has to be up like 98, 99, 100%. And then you maintain physiological conditions in the respiratory fluid or whatever the fluid is, that original droplet. And if the virus was happy before, then it's happy now and can survive. So to summarize, I've talked about how there are widespread sources of viruses and, as we know, other microbes in the built environment, in getting into air at least. We know that we've shown airborne viruses are subject to aerosol dynamics, just like any kind of abiotic particle. We're working more to dissect this relationship of how virus stability depends on humidity. We're interested in trying to determine the mechanisms for that. And I think some of the future challenges that we face are viral metagenomics, especially in air. We have the biomass problem. Could we possibly get enough? We've tried filtering out the bacteria, but I think viruses are probably stuck to bacteria. We may be losing them. There's a whole host of challenges there. Will we get? I think we'll get there eventually, but it's a big issue right now. I think the interactions between viruses, bacteria, and fungi are very interesting. That's more kind of an ecologist question, but I think viruses are important for transferring genomic material between microorganisms. And then really understanding the virus or bacteria or fungi microenvironment, I think, is important for determining the viability and the activity, metabolic products, and other things that we like to talk about. And then the final slide here shows some of the references that I've discussed today. So I'd be happy to take any questions, I guess, after Dr. Taylor goes. Thank you.
Thank you very much. Just as we did yesterday, we'll open it up for discussion after both of the talks. So it's my great pleasure now to introduce someone I've known for longer than I'd like to admit, John Taylor from University of California at Berkeley, who will speak about my favorite life form, fungi in the built environment. Go, John.